Good afternoon. Today we're gonna explain you how we're gonna overexpress recombinant proteins in Saccharomyces cerevisiae.
Well, the contents of node projects are the justification, introduction, another time we are going to explain justification, hypothesis, metabolic engineering strategies, and conclusions.
The production of recombinant proteins is an industry that is growing every year, and high amounts of proteins can be obtained from engineered organisms that are a better alternative than natural sources, because often these sources provide limited and low concentrations of the product, and the obtention is more expensive. It is important to take into account the selection of the host. In this case, it is Saccharomyces cerevisiae, and it presents many advantages over other organisms. It has fast growth. We know it's completely known. It is easy to manipulate, and it can perform post-translational modifications. For example, the formation of the disulfide bond that is important for the secretion of the proteins.
This organism has been recognized since 1982, its whole genome, because of the production of insulin, which started the boom of recombinant protein production and cell. And it's also a grass organism.
As a part of the justification, we have another characteristic that is important characteristic that Saccharomyces cerevisiae have, that it is important for us, the humans. For example, it has been used as a part of model of the human diseases at the beginning of the study of these organisms. But when they, as Isabella says, found that it have a potential to produce another important protein recombinations, but they start to making these proteins in their examples like insulin, another parts necessary for us. There are many other examples of model organisms, but the most in eukaryotes, most important are Pichia pastoris and Pichia and Conducella polytrophia. As we are saying, it's well known this organism since the beginning, because with this we have another important product. In the past, the modifications that were made were only as a random modification. So we didn't know how that modification that we did, how they affected. But now, with the strategies, with more sophisticated strategies, we know how to do in what part is important to these modifications.
Also, it's important to recognize that these enterprises of pharmaceuticals have a gross net income of $2,000 billion in 2011. And it's also have been prognosticated for the year of 2018, a growth of $32 billion for this industry. Also, it's important to know that this organism has the ability to make post-translational modifications, which are the well-known glycosylations down to proteins. And also, it's the factor of 20% of the whole biopharmaceuticals recombinant proteins in the whole industry. Finally, we know that we have positive strategies, but why do we have to make metabolic engineering? Well, firstly, we have a problem among production of recombinant protein and cell growth. And it's a competition, and also that the pathway of secretory pathway of proteins requires a lot of energy, and most of the energy is implicated on cell growth. So the main goal for us was to design a metabolic engineering strategy in order to overproduce recombinant proteins in Saccharomyces cerevisiae by changing the carbon flux.
We found in literature many ways to increase this production of proteins by engineering genetics. So we thought that redirecting the carbon flux toward the production of biomass and energy will increase or will optimize the production of proteins. So we found specifically for Saccharomyces cerevisiae many ways to do that, and the strategy will focus on increasing the power glycogenase production and also the nucleic acids and amino acids production. So first we found this pathway that is glycolysis and gluconeogenesis by KEGG, and we start from the principal enzymes used in Saccharomyces cerevisiae. This in order to have an approximation of what modifications or perturbations we have to do, and what another pathways are just taking energy and that diminish the production of proteins. So first, we propose the utilization of a mixture of galactose and ethanol as a substrate. This will be possible using an strain that contains GAL10 promoter. And also, we propose the knockout strain of FBP1 and PSK1 genes. This in order to delete the pathway of fructose, that it will be unusable. So we found also that in the production of proteins, the galactose and the mixture of it with ethanol increase the pentose phosphate pathway activity and TCA pathway activity. So it is a good approximation for recombinant protein production. And here there are the two enzymes that we will knock out, and these will not disturb the rest of the pathway. So we're gonna overexpress also pentose phosphate pathway in order to increase the production of glyceraldehyde, and this will be the source for the next pathways. So another strategy that we are proposing is the overexpression of aldehyde dehydrogenase. That is an important producer of NADPH in yeast, as we know by places. So we found also that NADPH is required for the synthesis of amino acids, and this will be accomplished with the addition of a promoter upstream of the gene AL6. And next we can found this enzyme that is interconverting acetaldehyde and aldehyde. So it will increase the production of NADPH, and this is an important source of energy.
Well, another strategy is the inhibition of the alcohol dehydrogenase in order to delete the fermentative pathways, because it removes the carbon flux. It uses carbon flux to create ethanol instead of TCA and also acetate. So with this modification, we can redirect the carbon flux towards TCA in order to have more energy and power reducing agents, and also acetate, as we already said, is one of the main sources of NADPH. And this can be achieved by the knockouts of these genes that codes for the alcohol dehydrogenase, that are the ADH1, 2, 3, 4, and 5. And also, because we are using galactose and ethanol a culture medium, it's been observed that in the presence of ethanol, the synthesis of the alcohol dehydrogenase is inhibited in a natural form. So this will also cause the deletion of this pathway, and also it has been observed that with galactose, yeast perform a better respiration, so it also inhibits the fermentation.
Okay, because of all the carbon flux that is going to be received on TCA cycle, we must also increase the activity of the enzyme that transforms pyruvate into oxalacetate. This enzyme is the pyruvate carboxylase, and it has been reported that with zinc cofactors, this enzyme has an increase in activity. This would be an external way to direct or to increase the activity of this enzyme. Another way would be to overexpress this with a strong promoter. And this would be the enzyme that will be interconverting pyruvate into oxalacetate.
So the next strategy is overexpress pentose phosphate. As we know, this is an important source of energy or power producing agents, specifically NADPH, and also is an important source of pentose sugars, and this is important because of the amino acids will be created from this source, and also modification of the pentose phosphate pathway has been reported as effective in fungal biotechnology. So we have information about the radio that that says that pentose phosphate pathway is decreasing Ender-Meyer-Parnas pathway. So it will be redirect the carbon through this pathway, and the production of recombinant protein is based on this. Also, we're gonna achieve this by the overexpression of 6-phosphoglucurolactonase and by the SOL3 gene, and this has been reported like the gene most effective for the overexpressed pentose phosphate pathway, and this has been achieved 40% increase in the producing of recombinant proteins. And here we have, these are yellow ones are enzymes proposed like possible targets to overexpress and to increase the production of the activity in pentose phosphate, but the red one is the most effective reported in literature.
Well, also because we need an increase in energy, we all know that the malic enzyme is a really important source of NADH and of NADH in cytoplasm. So in order to increase this, we shall change the carbon source. It has been reported that this enzyme is really sensible to glucose. It reduces its activity in 85%, while with galactose it has only a 67% of diminished activity. So this would be an alternative to stimulate the activity of this enzyme and give more power-reducing agents in order to create more proteins.
Well, putting together all the strategies you already mentioned, we build a pathway, the complete pathway in Metacy, and it starts with the degradation of the galactose using the Leloir pathway here until glycolysis. And we did the pathway of fructose, and we directed this towards the pentose phosphate pathway. Well, then here it goes to pyruvate, and here is the other modification we perform by deleting the fermentative pathway to the production of ethanol, and here we'll also redirect the flux towards the production of acetate. And also here it is the TCA that also this modification will redirect the flux towards the TCA cycle. Well, here is the malic enzyme, but the program didn't allow us to put this reaction, but here it will be another modification of the production of malate to pyruvate that will produce NADPH.
Also to add, this modification is really important because the fermentation is known because it's independent from acetyl-CoA and uses pyruvate. So when you delete dehydrogenase, you're adding a pathway that is creating more power-reducing agents by producing acetate.
So, as a conclusion, we have that the demand of the energy in Saccharomyces cerevisiae can be supplied with metabolic modification strategies, in contrast, and only increasing the source of the saccharides. As we saw in the class, we can modify any organisms to make with other carbon source to make or recombinant protein. Another metabolic pathway are part of the network. Perturbations will affect the metabolism, so we have to know with this perturbation we will know that our protein recombinant will be done, and with these modifications can be reduce the cost of the production of these proteins.
Well, do you have any questions?
I have a question.
Yes.
Maybe for this strategy that you presented us, how did you decide which enzymes will you shut down?
Are you talking about fructose?
Yes, yes.
That's because we select galactose has carbon source and ethanol, so fructose is not gonna be metabolized. So
It's useless.
we can exactly just use energy that you need for the protein production,